The complement system, or complement cascade pathways, aids or complements the ability of antibodies in phagocytic cells to clear pathogens from an organism. Though the result is always the same, there are three distinct initiation pathways. First documented in the 19th century, it has since provided one of the bases of humoral adaptive immunity and has spawned countless studies which have been attempting to fully understand its role in innate and adaptive immunity, and also its role in autoimmune disorders.
An antigen-antibody complex is formed on the surface of the pathogen. C1q, which is a subunit of C1, attaches itself to the antigen-antibody complex, and C1R catalyzes its own activation and that of C1s. Deactivated C1s then cleaves C4a from C4, leaving C4b unstable, which immediately binds to adjacent proteins or carbohydrates. Surface-bound C4b now binds C2 to become C4b2, and in the presence of magnesium ions become deactivated C4b2a. The C4b2a activated complex is the classical pathway C3 convertase.
The alternative pathway is also known as the antibody-independent pathway. The antibodies on the antigen would send away signals to attract the complement proteins. C3 complement protein undergoes hydrolysis to divide itself into C3a and C3b. C3a diffuses away slowly and leaves the complex, and C3b recognizes the antigen as it can bind to the foreign particle. The C3b fragment would get inactivated by the inhibitory protein in our cell if C3b gets bound to our own cell, as it cannot carry out further reaction. The C3b is activated when it is bound to the foreign antigen, and factor B would attach to C3b, and factor B would get cleaved by factor D. Factor B is bound to C3b, and factor D is bound to BA, which also diffuses away from the system, and BB travels to the target site. C3b, BB, also known as a C3 convertase, is an activated complex. C3a diffuses away, and C3b goes to the target site. C3b, BB, 3b, also known as a C5 convertase, acts on C5, and C5 divides into C5a and C5b. C5a will diffuse away, and C5b will elongate the complex, and the complex will be C3b, BB, 3b, 5b, and C5 will be the attachment site for C6, C7, C8, and C9, and this is known as the MAC formation.
Activated MASP2 cleaves C4 to C4a and C4b. Some C4b binds covalently to the pathogen surface. Activated MASP2 also cleaves C2 to C2a and C2b. C2a binds the surface C4b, forming the classical C3 convertase C4b2a. C4b2a binds C3 and cleaves it to C3a and C3b. C3b binds covalently to the pathogen surface. C3b causes the opsonization of the pathogen and cytolyzes via complement protein C5 to
The complement system is part of the immune system that enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells from the organism. It promotes inflammation and attacks the membrane. The proteins circulate in an active form, but in response to the recognition of molecular components of microorganisms, they work in a cascade where the binding of one protein promotes the binding of the next protein. Functions. Phagocytosis occurs by opsonizing antigens. C3b is the most important opsonizing activity. Inflammation occurs by attracting macrophages and neutrophils. Membrane attack occurs by rupturing the membranes of foreign cells. There are three complement pathways that make up the complement system: the classical pathway, the lectin pathway, and the alternative pathway. These pathways differ in the manner in which they are activated and ultimately produce key enzyme called C3 convertase. Classical pathway cannot act independently. It relies on the actions of B cells to produce antibodies specific to different pathogens, as well as other components of the innate immune system, such as macrophages. The lectin pathway is activated by the interaction of microbial carbohydrates with mannose-binding proteins in the plasma and tissue fluids. It is homologous to the classical pathway, the only difference being the starting process. In the lectin pathway, it is activated when it recognizes sugars on the microbial surface. So if a bacteria that enters the system has a sugar on the surface, such as mannose, the system will recognize this and start the lectin pathway. The alternative pathway is activated by C3b binding to microbial surfaces and to the antibody molecules. The pathway is triggered when a C3b protein directly binds to a microbe. It can also be triggered by foreign materials in damaged tissues. It cleaves C3 until there is enough to continue the steps of the classical pathway from the C5 convertase step. The end results and events benefits of each pathway are the same. All pathways carry out beneficial innate defense functions. They trigger inflammation, chemotactically attract phagocytes to the infection site, promote the attachment of antigens to phagocytes, play a role in the activation of naive B lymphocytes, cause lysis of gram-negative bacteria, and remove harmful immune complexes from the body.